this is Monica Reddy. Today I would like to talk about microbiology and physiology chapter, that is 1.3, cell classification of bacteria. Bacteria are classified depending upon three cases, that is shape, gram staining and oxygen demand. As we all know, bacteria are multicellular organisms. They are ubiquitous everywhere. They are present everywhere. In hot thermal conditions, in every conditions, they are present. So, bacteria can be classified into various categories depending upon these three cases. Okay, these three conditions. Those are shape, gram stain, and oxygen demand. We all know the structure of bacteria, that is a simple bonded design. Bacteria are cell microorganisms, okay, with the absence of nucleus. So now let us know the classification. First, the shape. Shape, according to the shape, there are three types: spirilla, bacilli, and cocci. And coming to the gram staining, we have two types: positive gram stain and gram negative. And oxygen demand. Under oxygen demand, we have aerobic and anaerobic. Aerobic is the presence of oxygen, anaerobic is the absence of oxygen. And the classification of bacteria is depending upon these factors. First, it is depending upon shape as well as staining, followed by action and the basis of oxygen requirement, and on the basis of pH, on the basis of temperature, on the basis of osmotic pressure. First thing, on the basis of oxygen requirement. Under this, we have five types, that is obligate aerobes, facultative aerobes, aerotolerant aerobes, anaerobes, obligate anaerobes, and microaerophiles. Okay, these are the type of bacteria. They are categorized into five types. These are obligate aerobes. For example, for this is Pseudomonas mycobacterium, and for facultative anaerobes is E. coli. Aerotolerant anaerobes is Streptococcus pyogenes. Obligate anaerobes is Clostridium bacteroides, and microaerophiles, Campylobacter. On the basis of pH, even in the conditions of pH, also bacteria will survive. Under that, they are classified into three types. That is, like we all know that bacteria are also very versatile organisms. They survive in extremely conditions, such as they are also called as extremophiles because they did survive in extreme conditions. Even they are called as extremophiles. They are divided into three types: acidophiles, neutrophiles, and alkalophiles. Acidophiles is nothing but under the condition of in the acidic condition also the bacteria is present. Okay, and neutrophils is nothing but under the neutral condition also bacteria are present. Nothing but Escherichia coli. And alkalophiles, even the condition is alkaline, the bacteria is present. So here is nothing but simply we can say that bacteria are present in every pH conditions. Okay. And the third case is on the basis of osmotic pressure. That is nothing but osmotolerant and halophiles. Some of the examples are given here. You can follow it. On the basis of shape. On the basis of shape, bacteria are divided into three types: cocci, bacilli, and vibrio. Okay. Again, it is in under cocci, under these three things, again they are subdivided. Okay, depending upon their shape and the arrangement and their like arrangement and their uh, yes, the first one is diplococci, second one streptococci, sarcina, tetrad, staphylococci. Under diplococci is nothing but two circular forms of bacteria attached, and strepto is nothing but many, and staphylo is a large cluster of group of coccus bacteria are formed to form a huge cluster, nothing but a staphylococci. Okay, there are many examples are given here. Coming to the diplococci, it is Neisseria gonorrhea, And for streptococci, Streptococcus mutans, and Sarcina rosea. And here some of the examples are given below for tetrad and Staphylococci as well. And the next one is bacilli. Bacilli are again divided into two types. We usually know that bacilli are rod-shaped or cylindrical-shaped bacteria. Diplobacilli is nothing but two cylindrical forms are attached. Cocobacilli is nothing but a cluster form of bacilli are attached, like cluster form of bacilli. Okay, we have we are given with many of the examples over here. Nothing but a Clostridium and Bacteria pertussis. In the last but not least, nothing but a Vibrio. Vibrio is nothing but a Vibrio cholerae. These are spiral forms which are irregular in shape. They are in coil structures. Okay, this spiral actually it's a very useful bacteria to the human. As we know, there are many astronomers who got up to the space. They'll have this spirulina as a supplement in place of food. They'll have as a supplement, because as it has a capacity to produce huge amount of energy. Spirulina has uh, a great it has a function that it produces a huge amount of energy when compared to proteinaceous food. So in the absence of food, they'll have spirulina for their survival in the space. And on the basis of pH requirement, okay, the I already told you that it is classified into estophiles, neutrophils, and alkalophiles. Like, as coming to the acidophiles, in acidic conditions also the bacteria are present. Like, they require very low pH. When we usually know, when the pH get down below uh, like um, low, like when the pH gets low, it is nothing but usually an acidic. Okay, even though it is acidic, there are many forms of bacteria will grow under acidic forms. Nothing but we have given with the examples, that is thiobacillus, ferrido ferrooxidans, can tolerate up to pH 1. See, it's very highly acidic pH. And flavobacterium species, like there are many species which can survive under very low pH. And there are some acid tolerant bacteria are lactobacillus and acetobacter. And neutrophils. Neutrophils are nothing but these are the bacteria which are highly, which like they can survive in neutral conditions with a pH of 6.8. Some of the bacteria like alkaligens, Pseudomonas, E. coli, etc. Alkalophiles are nothing but in alkaline conditions. Okay, that is nothing but their growth is seen, or they grow well in pH 8. That is nothing but a basic pH level. Is it? That's a basic pH level. Examples is Bacillus alkalophiles, Bacillus circulans, Natronococcus species. These are some of the examples for alkalophiles. Next one, osmotic pressure. Bacteria require 80% of water for their growth usually. So what they do is they create sufficiently hypertonic media. Hypertonic media is nothing but the concentration of solution is high. Like nothing but a high concentration solutions. So at that hypertonic media, the concentration will be greater than those inside the cell, cause water loss from the cell. Because of this, I already told you, like hypertonic is nothing but the concentration is higher. So due to this, because of the medium higher concentration, what it does is it makes the plasmolysis of the cell. It I mean nothing but it causes a water loss from the cell. Examples like osmosis, fluid leaves bacteria causing cell to contract and causes the cell membrane to separate. Plasmolysis, this is also another method, nothing but cell shrinkage. Extreme or obligate halophiles, adapted to and require high salt concentrations. Oxygen requirements. Oxygen requirements, in that we have four types: aerobes, obligate aerobes, macroaerophilic, obligate anaerobes. Aerobes is nothing but they grow under aerobic condition, which contains 21% of oxygen and very less amount of carbon dioxide, that is 0.03%, and obligate aerobes. They strictly require oxygen for their growth. Example, Pseudomonas aeruginosa and macroaerophilic bacteria. These are the species which grow under reduced oxygen content and 5 to 10% increased of CO2 and 8 to 10%. And last case is obligate anaerobes. They doesn't need any oxygen for the growth, but here the oxygen is inhibited. Simply nothing but. See, on the basis of strain, we usually know. Depend the main case like for staining technique is the presence of peptidoglycan cell wall, peptidoglycan layer in this cell wall in bacteria. This is present in both the positive and as well as negative bacteria, negative stain bacteria. So what happens is in positive bacteria they have thick layer of peptidoglycan, and coming to the gram negative we have a very thin layer. We have very thin layer. So what happens is gram negative bacteria will have a high lipid content in the cell walls, gram negative bacteria, and the alcohol, like we stain with the alcohol after staining with the counter stain, we stain it again, we wash with the alcohol. What happens is this alcohol will allows the leakage of crystal violet iodine complex. So that's what the gram positive bacteria with less lipids in their cell walls are less susceptible to the action of alcohol. And coming to this peptidoglycan layer, like these are formed in cell walls, as I already told you. In gram positive bacteria, we have thick peptidoglycan layer, as it traps more stain. As we have thick layer, they trap more stain, and negative one we have thin layer, so it has less peptidoglycan and fewer cross linkages, so they usually lose stain very readily, so they doesn't acquire stain very much. That's what gram negative species will be always visible in pale color. This is the main reason. And next slide. And the basis of classification. This is another type of classification. This is entirely related from to microbiology. This is a classification from microbiology, and that is nothing but a phenotypic classification and genotypic classification. Under phenotypic classification, we have under the subdivisions nothing but a morphological, anatomical, staining, cultural characteristics, nutrition, environmental factor, biochemical reactions, antigenic structure. and and genotypic class in genotypic classification we have DNA hybridization and guanine plus cytosine content. Let's discuss briefly. Under phenotypic classification, I already told you, depending upon the gram stain, shape, rod, spheres, etc. All this comes under this morphology. And macroscopic hemolytic properties on agar containing blood pigmentation of colony, size and shape of colony, smell, color, etc. And the third case is stereotyping, nothing but antibody reactivity to the specific antigens. And the fourth one is antibiogram patterns, susceptibility to antibiotics. And the last one is phase typing, susceptibility to bacteria and bacteriophages. And second one is genotypic classification. Genotypic classification is nothing but the comparison of nuclear material or inherited genetic things. Okay, genetic material, nothing but a comparison of genetic material, inherited instructions it carries within its genetic code. Similarly, between two organisms, individual genes or whole genomes can be compared. 70% homologous belong to the same species. In the like they'll be comparing with the they'll be comparing in the genomic terminology, like in the form of genetical information. With the help of the genetical information, they are classified depending upon the DNA, RNA, as well, every like there. And the shapes of bacteria. We have three shapes of we have three types of bacteria classified depending upon their shape. Nothing but a cocci, bacilli, and the last one is Vibrio, Spirilla, and Spirochetes. Okay, cocci are nothing but the spherical form of bacteria. Examples for them is Staphylococcus pneumoniae. In cocci, depending upon the arrangement and occupancy of spherical bacteria, they are again divided into three types. Okay, not three, it's four: Diplo, Strepto, Tetrad, Staphylococci, and Sarcina. Okay, Bacilli are nothing but it's a cylindrical rod-shaped bacteria. These are also attached in some cases, nothing but Diplobacilli. I already told you in the last slide. And for some species in bacilli, we can found flagella even. That is nothing but a salmonella typhoid, which causes typhoid. And the third one is spore former, nothing but the Clostridium botulinum species. Okay. And the others are nothing but Vibrio, Spirulina, and Spirochetes. These are the other forms, okay, which are irregular in shape. And cultivation of bacteria. Why we have to cultivate bacteria? What is the necessary of cultivating bacteria? See, we all know bacteria are ubiquitous. They are present everywhere, all over the any extremity. They can like they are present everywhere like that. So first thing, why we have to cultivate is like we have to observe the growth of the bacteria under laboratory conditions. What is laboratory conditions? Is nothing but growing the bacteria in vitro conditions, like giving a proper environmental and physiological conditions to the bacteria, like temperature, enrichments, etc., etc. So all this to be provided under laboratory conditions. Okay. And usually some properties of metabolic activities, virulence, change of genetic information, incorporation, all these can be observed under cultivation. And biological material. Biological material is nothing but striped on the plate or inoculated to the cultivation medium, solid or liquid for multiplication, thick, dense growth. See, we usually do a streaking and inoculation just to observe the growth. That's all. And for isolation even. And bacterial cultivation methods. We have two types. How we culture bacteria, of course, it's usually in laboratory conditions. That is, in the with the help of two substrate mediums, nothing but agar as well as broth medium. Agar is nothing but it's a semi-solid form, and broth is a liquid culture. And like in broth, we can directly inoculate our specimen using the cotton swab or plastic pipette. But on coming to the agar, it's a semi-solid form, as I already told you. We have to streak it. We have to follow a spread plate method, pour plate method, like that. That's all. These are the commonly used methods in agar plate method. Okay. And the purpose of culturing. What is the main purpose of culturing? Like isolation of bacteria, properties of to know the properties of bacteria, like that is culturing bacteria is the initial step in the studying its morphology and identification, as I already told you, to observe its internal structure as well as external structure in its functions, identification. For all these purposes, the bacteria is cultured. And maintenance of stock cultures for several years. Like there are some species where very hard to culture them. For that, in such cases, this stock cultures is used. And estimate viable counts to test antibiotic sensitivity, to create antigens for laboratory use. And certain genetic studies and manipulations of the cells also need a bacteria to be cultured in vitro. Culturing on solid media is another convenient way of separating bacteria in mixture. And as I already told you and discussed about all these purposes, it is given in the form of flow chart over here. In the next thing is bacterial reproduction. As we all know, In bacteria, reproduction is reproduction is carried out through a asexual mode of through asexually. Yes, bacteria usually undergoes reproduction through a asexual mode of reproduction, called nothing but a binary fission. Nothing but in this, a single cell bacterium divides into two daughter cells. These are identical to the parent cell as well as to the each other. Okay. What happens is, like they are identical to the parent cell as well as they are identical to each other. Replication of DNA within the parent bacterium. Then the first cell, we can see in the first cell that replication is taking place within the parent bacterium. That is nothing but the initial stage of division or a fission. Eventually, and the cells get to elongate, and there is a central ring formation that leads to the formation, that leads to the eventually cell elongation and to form a two daughter cells. And usually the rate and timing of reproduction will be dependent upon the conditions like temperature and availability of nutrients, as we all know. When there is a favorable condition, E. coli will produce its more population, like it divides very huge, like very fastly, and we can observe a rapid growth. Another diagram for binary fission. First case, cell wall, plasma membrane, nothing but a DNA nuclear material where the replication takes place, and the cell wall plasma membrane began to divide, and the cross wall, nothing but a ring or wall, whatever, that forms in between two cells, parent cells, that leads to the division, and the cells get separated with their own inherited chromosomal materials. And bacterial reproduction is, of course, strictly asexual, but undergoes sexual reproduction in very rare cases, like genetic recombination in bacteria has a potential to occur through. We have many recombination methods like conjugation, transformation, and transduction. We already know them very well. In such cases, the bacteria may become resistant to antibiotics. With the help of this recombinant methods, the bacteria gets very much susceptible or resistant to antibiotics. And there is a variation in the genetic material as well. Yes, we get the genetic variation. That's what. And bacterial growth curve. The entire growth cycle of a bacteria is represented by this graph. This graph shows the following four phases. It is like this is the graph which is drawn in between the time and the growth. Okay, time and the number of bacteria is nothing but a growth per time. First one is lag phase, followed by log phase. Log phase is nothing but an exponential or logarithmic phase, stationary phase, death phase. Lag phase is nothing but the initial stage or the initial phase of the bacteria, where simply in simple terminology, we can term it as it's an infant stage where the bacteria uh, gets new to its environment, and they get adjustable to that provided environment over there, where it is completely inactive over there, where we cannot observe the division even. It's just like a resting phase, starting initial, not resting, initial phase. And then followed by the log phase. First one is lag phase. I already told you. They just adapt the growth conditions. It is a period where bacteria gets mature, not but they doesn't divide. I mentioned they don't divide or doesn't divide in the lag phase. Okay. And during lag phase, we can see the synthesis of RNA, enzymes, and other molecules in it. Okay, and this phase lasts for 1 to 4 hours, but the time duration is different for every species of bacteria. Okay, we have different time for every species of bacteria. And the next one is log phase or exponential phase, sometimes called the log phase or the logarithmic phase. which is a period characterized by cell doubling. Nothing but I already told you here, the division takes place that results in the rapid growth of bacteria, which doubles each cell, like cell doubling. The number of new bacteria appearing per unit time is proportional to the present population. I mean, like when the time increases, like per unit time, the growth of bacteria, like not growth. The division of bacteria will be proportional to the present population which is present in the media. Are you getting? So this is what the growth is not limited here. Okay, the growth is not limited. The doubling will continue at a constant rate. So both the number of cells and the rate of population is gonna increase in this phase. Okay, this is nothing but exponential growth stage. This is what it is compared with logarithmic phase. And stationary phase, I already told you, stationary phase is nothing but a stable phase where where we cannot see the growth as well as division, anything. It's completely stable over there. So in this stage, the microbial depth is equal to the microbial growth. So depth is equals to growth. అంటే like there won't be any change. It's completely resting phase, followed by death phase, of course. Death phase is nothing but die-off of cells called death phase. It is a period which the cells are dying at an exponential rate. So the growth which is present, like the growth which is present before death phase, is equal to the death phase now, like exponential rate. Some reasons are because of accumulation of what are the reasons for this death phase is accumulation of waste, loss of cells' ability to detoxify toxins, etc. Thank you. And I would like to continue with the continuation of this topic. Nothing but preparation of bacteriological media. This is a very essential topic in microbiology, as we all know. Preparation of bacteriological media. What is bacteriological media? Bacteriological media is nothing but a artificial media or a place where it allows the growth of bacteria when it is compared when is compared as of it is grown in the outer environmental conditions. The same environmental factors like temperature, nutrients, physiological condition, all will be provided in the media artificially. Nothing but a bacteriological media. So these are the some few images regarding culture media. We all know that bacterial cultural media or culture media is crucially, and firstly it is divided into two types actually. uh, Put it aside first, put it in your mind that in the first case actually it is divided into two types depending upon the consistency, like solid and liquid. So coming to the solid, we have agar, and liquid, that is nothing but a liquid broth. So, in solid medium, the media is nothing but agar. It is useful to study the colony character, and it is also useful for identifying mixed bacteria. Okay. Mixed bacteria can be separated in this solid culture media easily, very easily it can be separated. Solid media is used for the isolation of bacteria for pure cultures. For pure cultures, this solid media is firstly selected, and agar is most commonly used for preparing solid media. We all know agar is a polysaccharide form. It is obtained from the seaweed, and it is a solidifying agent as well, and bacteriologically inert. And coming to the, yeah, it remains as a solid at 37 degrees Celsius, and it is transparent, as we all know. Liquid media, um, liquid media is like it is used to observe the profuse growth of blood culture in liquid media. Mixed organisms can be observed cannot be. Now, now coming to the crucial classification, nothing but depending upon three cases: physical, chemical, and special purposes. Coming to the physical, I already told you consistency, nothing but liquid, semi-solid, solid. Okay. And coming to the chemical, synthetic and non-synthetic. And for this, and coming to the third case, special purpose, nothing but the basal, enriched, selective, differential, anaerobic, transparent, antibiotic sensitive. First one. Next slide. Nothing but a basal media. This is a media where the bacteria grow that doesn't need any enrichment of media. Like it is provided by itself. The media itself, it is provided. We shouldn't add any kind of additional things to it. Like examples for this are nutrient broth, nutrient agar, peptone water. And in this basal media, a few species of bacteria are selected for growth. Example, Staphylococcus enterobacteriaceae species will grow in this media. And the next slide, enrichment media. This enrichment media, I told you, additional to the agar medium, we have to add some additional substances to it, uh, to the growth media. Like usually we add additional substances like blood, serum, egg, something like that, or else alkaline peptone water, nothing but APV, we call it in a short term. Alkaline peptone water also can be added for more enrichment. And for enrichment media, streptococcus species will grow in this enrichment media. Blood agar, Lowenstein, Jensen media are also best examples of enrichment media. And the next slide is selective media. I already told you selective media is nothing but only few selective species of bacteria are grown in this type of medium. This media favors the growth of a particular bacterium by inhibiting the growth of undesired bacteria. Only the bacteria which we need to cultivate will be only grown in this type of bacteria. The other species, like the other guest coming to this media will be not allowed by the desirable bacteria. Are you getting? Yeah. This is what happens over here, allowing the growth of desirable bacteria. Examples are McConkey agar. McConkey agar is the best example of this, and Lowenstein Jensen medium. I'll deal with a little example for this. For selective medium, actually they have some of the scientists have conducted experiment, like they have grown some of the throat microorganisms like Neisseria. Okay, Neisseria in this selective media. So what this Neisseria did is it did not allow any other forms of bacteria. Like of course it belongs to the throat portion, but it did not allow the other throat microorganisms to be grown in the selective media. Like only here the profused growth of Neisseria bacteria is only seen, but uh, but the rest of the media, but the rest of the growth of microorganisms are absent over here. They have concluded that the diphtheria causing bacteria are not grown in this bacteria. So for this media, we can also add antibiotic for inhibition. Okay, and this is also, and tellurite media is also best example for selective media. And differential media are also called as indicator media. Why we call as indicator media is we add indicator to the medium because a particular organism causes a change in the indicator that changes the color or its color when bacterium is grown. Example, uh, blood neutral red, and tellurite is nothing but a indicator. Okay, blood agar and McConkey agar are the indicator media. Next one, differential media. As I told you, more than one type of microorganism grown on it. Many type, more than one type ante we have multiple types of bacteria grown on this differential media. For their identification, particular identification, we add indicator to the differential media. Okay, Transport medium. This is a medium used for transporting the substances. Okay. So, like these media are used when a specimen cannot be cultured soon after collection. Example, Carrie Blair medium and amino medium, Stewart medium are the examples for transport medium. Actually, it's a organic, simple organic medium which maintain the viability of all organisms in the specimen. And transport medium inhibits the self-destruction and multiplication of microorganisms. So the growth will be a stable. There will be no increase in growth as well as there will be no decrease in growth. Okay, stable గా ఉంటుంది. So some delicate organisms like Neisseria, Haemophilus, Shigella will be grown in this transport medium. Only few delicate species will be grown in transport medium. Next slide. Anaerobic medium. Anaerobic medium is nothing but anaerobic bacteria need a special bacteria for their growth or for their survival, whatever, for growth, because they need low oxygen content, nothing but reduced oxidation. It have nutrients like hemin, vitamin K. Example, thioglycolate medium is the best example for this, and Robertson cooked meat medium. You can see this Robertson cooked meat medium. Here also we couldn't see the presence of oxygen over here. That's what this anaerobic media is used for the cultivation of anaerobes. And the antibiotic sensitive media. This antibiotic sensitive medium, actually it's an assay, is made to determine the ability of an antibiotic to kill or to inhibit the growth of living microorganisms. This is what happens in antibiotic sensitive media. And these are the common ingredients of a culture media: peptone, beef extract, yeast extract, agar. From peptone, we can get the source of carbon, nitrogen as an energy. Okay, and from beef extract, we get amino acids, vitamins, minerals to the medium. And from yeast extract, we get vitamin B, carbon, nitrogen sources. And the last but not least, agar, it's a solidifying agent. And coming to the next one, staining. Why we have to do staining? We have to persist staining because in order to differentiate between both the gram positive as well as gram negative bacteria, we have two types: simple staining and differential staining. Simple staining is nothing but a use of a single stain, nothing but for visualization of morphological shape and arrangement, nothing but the outer structure, to find out the outer structure and morphological shape and arrangement of bacteria. This differential staining, I already told you, for identification and visualization of structure clearly, okay, internal. So under identification, it comes like gram positive and as gram, like to identify gram positive and gram negative bacteria. And for this visualization, under visualization, we can observe spore staining and capsule staining. First, coming to the first simple staining method, actually, okay. First, I'll discuss about gram staining. Gram staining, it is firstly reported by a Danish scholar. It was discovered by a Danish scholar named Christian Gram, and it is named after his name. Okay, nothing but a gram staining. Okay, in 1884, he discovered this gram staining technique. First of all, a smear of bacterium is taken. To that, a crystal violet is applied for 30 seconds and then slide rinsed with water, washed thoroughly with water, and the iodine is applied and it is also rinsed off. Then we can see the then we can see the absorption of color to the bacteria. We can see that pink color, nothing but a saffronin. We added here, actually we can take saffronin as well as crystal violet. So we can see gram positive bacteria are in violet color and gram negative bacteria are in light pale pink color. So this is what happens in gram staining technique. This is what happens in gram staining technique. And the next one, gram positive and gram negative. As I already told you, first of all, you have to fix the bacteria, you have to add a crystal violet, wash off, and then followed by iodine treatment and wash out. Then it leads to, of course, decolorization. And then you have to add a counter stain, nothing but a saffron in to color the negative species as well. So this is what happens in um, gram staining technique. Acid fast staining. Acid fast staining is it's a technique where it is applied to mycobacterium species. Okay, they're difficult to stain by ordinary methods. Usually, these species like mycobacterium, they are very difficult to stain. It seems so. That's what for them acid fast staining technique is employed. Okay, so first thing is in this, the heat is applied. Heat treatment to be applied to this at acid fast technique. What happens when we apply heat to the like to the bacterial slide? The dye molecules go through the cytoplasm. Once stained, however, cells not easily get decolorized. Even when alcohol solutions containing 5% of acid, the organisms are said to be acid resistant or acid fast. And here, for acid fast staining, we use a stain nothing but a Gill-Nielsen carbon fusion. We use Gill-Nielsen carbon fusion. We use the stain named as Gill-Nielsen's carbon fusion. What happens is the cells become bright red. Then the smear is rinsed off with alcohol. Nothing but it becomes decolorized, and then the acid fast. What happens is the acid fast bacteria remain red, while the other bacteria lose their color. This is what happens in acid fast staining. And the counter stain, here we use counter stain nothing but a methylene blue is applied to the decolorized bacteria. Methylene blue. After counter staining, after counter staining, we add methylene blue. Nothing but in counter stain, we add methylene blue to the decolorized bacterial species. Okay, and again it is also rinsed off, dried, and examined. This is a technique useful to useful for the diagnosis of some disease causing bacterium like tuberculosis bacterium. Okay, and we can also add tergitol, nothing but it's a lipid detergent. We can add to the stain, nothing to the carbon fusion. Okay. Next, acid fast staining. I told you no. See, we'll add carbon fusion, application of heat, application of alcohol, decolorizer, and then counter staining. So after taking counter stain, the acid fast bacteria will acquire color, and the non-acid fast bacteria will be in blue color because we have taken methylene blue on the blue. See, acid fast is in colored, and non-acid fast is in methylene blue color. Blue color. Okay. This is nothing but a spore staining. Spore staining is also a special technique where spores are stained by applying heated malachite cream. Here also heating treatment is taken place, and after vigorous washing with the water, the stain from vegetative cells. Then safranin is applied to the smear. So what happens when safranin is applied? The technique observes the green spores and vegetative cells. For staining capsules, two principles are applied. First, a negative stain is done to the outline cell and its capsule. Next, simple stain. This is nothing but an endospore staining technique or a spore. Capsule staining. For capsule staining, we use here um, For capsule staining, we use a stain to the outer line of the cell for only for the capsule, and the simple stain is applied to the bacteria. For simple stain, we take safranin, and this results in the differentiation of the capsule as well as in the internal structure. What happens is the result of bacterium with the colorless capsule on the background, dark background. On the dark background, they appear colorless, colored with colorless capsule. Some of the gram-positive bacteria, Staphylococcus aureus, Corynebacterium, Clostridium botulinum, Micrococcus luteus, Bacillus anthracis, Streptococcus pneumoniae, Pseudomonas aeruginosa, Streptococcus pyogenes. These are some of the examples of gram-positive bacteria. And the next are gram-negative bacteria. Neisseria, I already told you, they're very delicate species. Spirillum, Escherichia coli, Treponema, Helicobacter, Neisseria, Vibrio cholerae, Streptobacillus maniformis. Some of the gram-positive and gram-negative bacteria. What is the major difference between the gram-positive and gram-negative bacteria is because of the formation of peptidoglycan layer, which is found in high concentration in the cell walls in positive bacteria, which traps the stain. Okay, which traps the stain, nothing but a crystal violet iodine complex, and coming to the negative bacteria with less peptidoglycan layer and fewer cross linkages, which loses strain readily. So it shrinks of the strain. It doesn't accumulate strain. Negative species. That's what they appear in pale color. Last but not least, staining techniques. Sorry, sterilization methods. Why do we have to sterilize things? Is it necessary to sterilize things? Yes, of course, it is very much necessary to sterilize things. In sterilization, we have three types: physical, chemical, and physical. Coming to the sterilization methods, physical. Physical is nothing but sunlight, heat, vibration, radiation. Coming to the dry heat, red heat, flaming, incineration, hot air oven, infrared. All these are the examples of dry heat sterilization. And moist heat, below 100 degrees Celsius, nothing but moist heat. Excuse me. No direct heat is applied over here. And radiation, non-ionizing, ionizing, electromagnetic particulate radiation. Okay. Filtration, we have earthenware, asbestos filtration, sintered glass and membrane filtration. We have these many types of filtration. And these are the types of disinfectant: air disinfectant, alcohol, phenol oxidizing agents. Disinfectants. There are many types of disinfectants. What is disinfectant? Disinfectant is nothing but a chemical which disinfects air, which prevents from infection. And air disinfectant, alcohol, phenol oxidizing agent. And here coming to the pasteurization method. Pasteurization method is also a type of sterilization which removes the bacteria, nothing but it's a pre-treatment of milk. In that pre-treatment of milk, we have 4 stages, nothing but a low temperature or long time pasteurization. That is, the milk is heated up to 63 degrees Celsius for 30 minutes, nothing but LTLT, nothing but low temperature, long time. For long time, low temperature, milk is heated. And next one is high temperature, short time, 72 degrees Celsius for 15 seconds. Okay. Ultra pasteurization. Ultra pasteurization is nothing but 138 degrees, very high temperature, only for 2 seconds, where we couldn't see the growth of bacteria. No growth of bacteria, no growth of lactobacillus. The next of ultra heat treatment. This is 140 for 1 to 4 seconds. This is also a type of sterilization method for milk. See, this is the sterilizer for milk. This is a machine which is used in companies for sterilization of milk. Nothing but a for pasteurization, ultra pasteurization, and all like that. And these are some of the chemical infectants such as halogens like phenols, iodine, chlorine. All these are chemical disinfectants which prevents from infections. Okay, which these are the halogens, chemical halogens, which is also a type of sterilization methods on inanimate things. On inanimate things, this is a sterilization on inanimate things for helping sterilization. Ethanol, propanol, alcohols are used for sterilizing the materials before culturing the things, and phenols like cresol, lysol, all these are phenols is also a disinfectant used for sterilization in laboratory conditions or for dismantling the culture media. And this aldehydes are also used, formaldehyde and glutaraldehyde, for preserving the cultures or specimens like that. And these are some of the precautionary measures while or not precautionary safety measures or cleanliness things to be maintained in a laboratory. First of all, you have to wash your hands, soap, take soap, rub hands, and the washing should be done properly in order to in order to avoid infections and infections and these are the lab safety rules. These to be followed in microbiology laboratories or biotechnology laboratories. Okay, in order to prevent from danger and danger and accidents. This is the usual dressing code to be followed in the laboratory. You have to. It's mandatory to have a lab coat followed by goggles, gloves, etc. This is an essential thing. Thank you.